Good afternoon, everyone. Myself, Mayank Roshan, a research associate at Functional Genomics and System Biology Laboratory, Animal Biochemistry Division, ICAR, National Dairy Research Institute, Karnal, Haryana, India. Today, I am here to present my research topic, Genomic Insights into Innate Immunity Variation among 6 cattle breeds, a comparative study using array CGH. So, this is the basic introduction. As we all know that India is home of wide range of cattle breeds, and each of them are adapted to the specific climatic condition and management practices. So, studying copy number variations is very important to assess how these different diverse number of cattle breeds are adapted to diverse climatic conditions. So, what exactly copy number variation is? Copy number variations refers to segments of DNA that are duplicated or deleted, resulting in variations in number of copies of segments of DNA. Copy number variation in genome can influence gene expression, phenotypic traits, playing crucial role in adaptability and innate immunity in cattle. Studying copy number variations may provide valuable insights into genetic basis of disease resistance and adaptation of different breeds. So this is the main aim of our research work. It is detection and investigation of genomic variation underlying innate immunity among 6 cattle breeds. And these cattle breeds were included in our studies where the indigenous cattle breeds, which are Sahiwal, Gir, Tharparkar, Kangayam, and 2 cross breeds. One is exotic breed, Holstein Frise, and other is Karan Frise. So this is the general outflow of our work, the methodology which we followed in our lab. So a total of 108 blood or semen samples were collected from 6 cattle breeds comprising 4 indigenous, Gir, Sahiwal, Tharparkar and Kangyam, and 2 exotic breeds, KF and HFs. The sampling was done at six animals for each breeds, and they were located at 3 distinct locations, each comprising of 6 animals for 6 breeds. A total of 108 samples were collected. So further, what we did was the genomic DNA was isolated, and we pulled all the DNA, genomic DNA into 6 sets of pooled genomic DNA, each comprising 3 subsets of pooled genomic DNA, resulting 8 pooled genomic DNA sample for each breed. The reference sample was fluorescein labeled with Psi3, which was HF. The test samples, the remaining breed indigenous and crossbreeds were labeled with Psi5. After performing the RSCGH, we went for self-to-self hybridization analysis of gene duplications and deletions and compared with reference Boss Torres genome ARS UCD 1.2. The chromosomal changes were further indicated by increase or decrease in fluorescence, and this intensity was measured with reference genome in terms of log ratio. The minimum log ratio was 0.25. After that, we filtered the amplification and duplicated genes and as well as gene deletions with the help of interactive Venn software. So after processing the number of amplified, duplicated or deleted genes, we further went for gene ontology or gene enrichment analysis study. The bovine gene list was utilized, and with the help of G-profiler, we segregated the duplicated and deleted genes. Further, what we did was we selected the innate immunity genes. The list of innate immunity genes were extracted from InnateDB software, and we further went for analysis of these important innate immunity genes through string software. We analyzed the protein-protein interaction network, and this fetched innate immunity genes were further uh, put in Cytoscape and CytoHubba software to find the important subgenes of each breed. So moving on to results and discussions. So this is the overall copy number variation among 6 breed. The left hand side of the Venn diagram and as well as the bar diagram depicts the gene duplications among 6 breeds. As we can see that the maximum number of amplification were observed in Kangyam breed. It was followed by Tharparkar breed, and very less number of gene duplications were observed in KF, HF, Sahiwal and Gir. So on the right hand side, when we see what were the results observed in deletions? We found very strange results as compared to duplications. In deletions were observed. We observed maximum number of deletion in Sahiwal breed. It was followed by Harparker, and rest of other breeds had very less or no number of deletions in their genome. So when we see the karyotype, how these duplications and deletions were observed among chromosomal locations, it was found that there was not any specific deletion and duplications. The deletion and duplications were non-randomly related to all over the chromosomes across 6 breeds. So karyotype with copy number variations showed these are the amplification. The red color denotes amplification. Then these are the deletions. So overall, the deletion and duplications were randomly duplicated over all the genome. So when we further went for gene enrichment analysis of duplicated genes, so it was found that in Tharparka and Kangyam, we had maximum number of gene enrichment analysis relating to molecular, biological, and other important functions. So in a nutshell, the important conclusions drawn from this gene ontology and duplications were that in Tharparker, the important pathway including the DNA metabolism, DNA binding, transcription factor activity, sequence-specific double-stranded DNA binding, and DNA templated transcriptions were observed. While in Kangyam, the biological process regulating mainly the L-ornithine transmembrane transport and important molecular functions associated with interleukin-1 receptor activity were highly enriched. We further observed gene enrichment in deletions too, but it was very contrary results were observed as compared to duplications in deletion. In deletions, we found that maximum number of gene enrichment analysis was observed only in one indigenous breed, that was Sahiwal, and it was followed by Harparker breed. While rest of the breeds we were not able to fetch any deletions. So in contrast to duplication, the results of gene deletion varied greatly, and mainly the gene involving the regulation of cell cycle and basic metabolism was deleted in Sahiwal, while in Tharparker the gene enrichment analysis show the MSC2 protein complex assembly was highly enriched. So as our research preceded the segregation and fetching of important innate immunity genes, so the amplified and deleted genes were fed into innate DB software, and we were able to fetch large number of innate immunity genes in different breeds. So the indigenous breed Tharparker and Kangyam had maximum number of gene amplification. A total of 76 innate immune genes were duplicated in Tharparker, while in Kangyam we found that 183 innate immunity genes were highly amplified. When we went for deletions, we only observed large number of deletions in indigenous breed Sahiwal. A total of 113 innate immunity genes were deleted in Sahiwal breed. So moving forward, what were the important hub genes that were important for providing any disease resistance to these breeds? So after fetching the important innate immunity genes in different breeds, we performed a hub gene analysis through cytoscape software, and we were able to segregate the important hub genes in Harparker, Kangyam, Sahiwal breeds. In Harparker duplications, we can see that these are the important hub genes. These are TGF beta 1, CCAM19, CD79, it is not possible for me to explain all the important hub genes, but TGF beta 1 is tumor growth factor. It is important for in cancer and apoptosis. CD79 is a marker gene which is important for B cell proliferation, and we have other genes too which are involved in, like AP821. It is involved in cell adhesion. Similarly, when we went for Kangayam, we too had some common genes like CD79A in both the breeds that were highly duplicated, and it was also found as hub gene TGF beta 1 interleukin 4, which is involved in cytokine uh, production in various cells, and cytokine production helps in proliferation of various immune cells. Notch is important in signaling pathway, and we have also some adhesion molecules which were highly modified, highly amplified. These were ICAM1 and AP21. But when we see for Sahiwal, we observe large number of deletions. These deletions were observed in these genes: MAP kinase 1, SMURF2, TRF1, and RAC1. All this, most of genes are involved in signaling pathway. So the overall conclusion of our research work is that we observed a large number of duplication in innate immunity genes in indigenous breed Tharparkar. 76 innate immunity genes were duplicated, while in Kangayam, a total of 183 innate immunity genes were duplications were identified. So we had very few number of deletions in Tharparkar and Kangayam. These findings suggest that Kangayam Tharparkar perhaps may have better adaptation to natural resistance to diseases in their region due to higher number of duplicated immune genes and absence of deletions in innate immunity genes. We are further carrying out experimental analysis to explore what are the important role of these hub genes and innate immunity genes for their survival in for the survival of innate for survival of this breed in indigenous conditions. So we would like to acknowledge ICAR NDRI, Shastra University, for providing infrastructure and support for research facilities. We like to acknowledge ISCB, Evolve CompGen COSI, for providing us opportunity to present our work in front of uh, you. We finally acknowledge our funding and agency NASF for support and assistance. Thank you.